Hello everyone, this is Roshan here to give a presentation on our work titled CoSMEL, a transformer-based approach to build context-specific metabolic models. Before moving on to what context-specific metabolic models are, let me give a brief introduction on what are genome-scale metabolic networks. So it is basically a reconstruction that collects all the relevant metabolic information of an organism, such as what are the reactions present in it. It also compares it in a mathematical model consisting of gene-protein reaction rules, which basically says what are the genes needed for a reaction to happen in an organism. It also contains a stoichiometric matrix where the rows correspond to the metabolites and the columns correspond to the reactions, and also constraints on the reaction fluxes. So basically a question arises here: do all the reactions defined in genome scale models are present in every human tissue or not? Well, the answer is basically no. Here's where context-specific model comes into picture. So it's basically a subset of the genome scale metabolic model defining a particular context. The context can be an organ, a tissue, or basically any disease. So traditionally, what is being done here to build a context-specific model is where we are using transcriptomics data and pass it through a thresholding method and map it to the genome scale model to extract a substrate using the model extraction method. But a problem arises here. This thresholding method is completely arbitrary, and there are no specific guidelines on how to do it yet. And based on this thresholding method, there are some housekeeping reactions that might be eliminated after going through the model extraction method. So our work, what we are using here is, instead of using transcriptomics data, we are using literature evidence and a transformer-based NLP model to build these context-specific models. So our hypothesis basically here is, given a set of abstracts pertaining to a specific context, it contains reaction information in them so that we can extract it using the transformer-based model. So our objectives are to build an automated pipeline consisting of context-specific literature extraction from a given keyword, basically, and predict the reaction present in those abstracts, and finally build a model using the model extraction method. And we also demonstrate the ability of this workflow by constructing a context-specific metabolic model of small intestine intrasite cell in the human organism, where we have given abstracts on small intestine intrasites. So moving on to the methodology here, we basically collect abstracts related to small intestine intrasites, pass it through the biointerest API, where we give the PubMed IDs and extract the relevant PubMed abstracts. And basically the transformer used here is known as BERN, biomedical named entity recognition and normalization. It is a multitask transformer model which can recognize up to 9 entity types. And based on these entity types, we basically collect all the enzymes, genes, and metabolites present in the PubMed abstracts. After collecting all the named entities, we map it to the genome scale model used here, that is Recon. And based on the gene protein reaction rules and the stoichiometrics, we map the metabolites based on Chebi IDs, genes based on the HGNC ID, and enzymes to the reactions based on fuzzy-wuzzy string matching. And we collect all the predicted reactions from the abstracts. After collecting all the active reactions predicted from the abstract, we pass it through the model extraction method to build a self-consistent context-specific model. These are the results that we have obtained. So basically, we have simulated around 100 random models containing where we have given random abstracts as input and built some context-specific models. So here we have around 100 random models, and the count indicates the number of models, and the x-axis indicate the common reaction present in them. As you can see here, our model performs around better than 95% of the models generated. And this is basically the fraction of the common reactions. And as you can see here, when we have used the init algorithm as the model extraction method, we have predicted around 2041 reactions pertaining to small intestine intrasites. Out of these, around 486 mapped with the original context-specific model for the small intestine intrasites. and our accuracy is around 51.43%. When we use the MCAT around algorithm, our predicted reactions are actually quite lower, and it also predicted only 146 SIC reactions, given accuracy of 15.45%. And moving on to the conclusion here, we have built an automated pipeline to predict reactions given abstracts as input. And comparing different model extraction methods, the unit algorithm basically gave us a better accuracy compared to MCAD. And based on the histograms, we basically validated our hypothesis that abstracts of the relevant context would contain reaction information in them. And so we are going to extend this pipeline by including where we give a keyword basically and extract abstracts related to that keyword using transformer based models. And also, in addition to NER, we're also planning to include a relation extraction as a additional task to better improve the performance of our workflow. Thank you.